The traditional T maze can be used to test both delay and effort-based decision-making. In the current work, we invented a T maze apparatus with automated food delivery, door management, and arm choice recordings. Our protocol here can dramatically save researchers' time and labor when analyzing the decision-making ability of rodents. This protocol can help to answer the key questions in decision-making fields, such as what are the key neurocircuits and molecules underlying decision-making procedure, and what are the neuropathological causes underlying decision-making deficits in psychiatric diseases.
The implication of this protocol extends toward therapy of neuropsychiatric disease because many patients demonstrate difficulties or deficit in decision-making.
Habituate mice to the maze for 10 minutes per day and totally for 5 days. Keep all the maze doors open during habituation. On day 1, scatter the food pellets throughout the maze. Then, place the mice in the start box of the T maze in groups of 4 and allow the mice to explore the maze for 10 minutes. On days 2 and 3, scatter the pellets along the 2 goal arms. Allow the mice to explore the maze for 10 minutes. Finally, on days 4 and 5 of habituation, put the pellets only very close to and at the food dispensers at the 2 goal boxes. Allow the mice to explore the maze for 10 minutes.